Welcome to this cryo-EM course. Last class I discussed little bit about the sample insertion and also how to use the objective lens as a focus and focus the electron beam. So, today we are going to discuss about the two other component, what is called the pump system in electron microscope and as well as the detector. That is, the two things are very important, but most of the time the pump system, even if we do not know or understand too much, still it is okay, but it is better to know how this pump system is working and why it is important in electron microscope. As well as the detector is very important, that how we can use those detectors and what is the importance of using the direct electron detector or CCD camera. So, different types of microscope we are using different types of detector also, and for the biological science sample, especially for cryo-electron microscopy techniques, detector plays a very important role, and that is the reason it will be good to know about the direct electron detector system as well as the normal CMOS camera or charge coupled device, how we can use those things the collect data. Before that, last class when I discussed about that defocus or focus, so mainly there are we are using objective lens system to focus any sample, and last class also I discussed little bit about that. Okay, we cannot focus where we are imaging the biological science sample, especially for optical microscope. Here wherever the target area, that if this is my target area, this is the we are targeting this particular finger, and if we want to target that, we will come here, we will focus this finger and then we will take that image. But for the cryo-electron microscope we cannot do this similar approach, because the most of the time electron has a very high energy, and whenever we are talking about cryo-electron microscopy technique, what do we have? We have the biological science sample embedded in amorphous ice, that I will discuss more, and that is going to melt or damage because of this high electron dose, and that is the reason most of the time what we have to do, we have to collect data or focus data in a different place, come back and collect data. So, I will discuss little bit, this is the part of the sample preparation. When we will discuss more about sample preparation, that time this will be clear, but today I will just draw a diagram and try to explain how that is happening, and same time this focus is very important, that why we are interested about this focus term, that is I am also going to discuss. Let us start. So, what do we have? Last class also I discussed little bit about this one, that when we have a grid square, grids means this is our grid, that is 3.05 millimeter is total diameter. If you just consider that this is the metal copper, so if you just look at this is copper made, and also in between that whatever you can see, those are the copper bar. So, it is a very tiny thin grid. So, you have to handle these things very carefully, you cannot do whatever we want. So, now if you look at one of these square, what do we have? One of these square, there is a uniform sizes hole inside that square, and these are the carbon area. Basically these are the carbon area. If you look one hole, how it will look from side? So, from side it will look like that. We have AFM data, that atomic force microscope data, where this will be carbon area, and this is literally a hole where electron beam can go through if there is no sample. Now, in the cryo-electron microscope, what we will do, generally we use a pipette, and generally a small amount sample, that is 3 microliter sample, we are adding into this or you can say that this grid, and then the sample will be distributed the entire grid uniformly. Then we are using a blot paper. So, we will use a blot paper to blot this extra buffer. This is, you can say, blot paper. So, this is, you can say, this is a blot paper. So, generally the blot paper will come from both the sides of the grid and blot the excess buffer solution. When there is a small amount solution will stay inside this hole. So, solution means it is basically carbon, hydrogen, nitrogen, oxygen, and solution means it is a buffer solution where we have the phosphate buffer, tris buffer, and then the pH will be adjusted. And why I told carbon, hydrogen, nitrogen, oxygen, that is our biological science sample. So, if we are dealing with a protein sample or a DNA sample or a RNA sample which is made by carbon, hydrogen, nitrogen, oxygen molecule, and it has a definite structure. So, those things will stay inside this as a solution. Now, what we have to do, we have to quickly plunge this entire thing into a liquid nitrogen, or you can say liquid ethane. So, liquid ethane, if I say that liquid ethane, okay. So, liquid ethane temperature is minus 180 degree centigrade is the freezing point. So, we are keeping temperature near around this temperature and quickly plunged into liquid ethane. When we will quickly plunged into liquid ethane, what will happen? There is a small amount of water buffer, whatever it is there in a solution condition, it will freeze quickly. Whenever it will freeze quickly, it will form an amorphous ice. So, amorphous ice, it may has a different shape, it can form a this kind of shape, or it can form a this kind of shape, or some other shape is also possible where our protein molecule will embed at different orientation. So, basically we are trying to image through this one, that electron beam will pass through this sample and give us a 2D image of a 3D object. So, this is the basic principle of cryo-electron microscope, where we are using this kind of grid, where we are using liquid sample, and then we are quickly freezed into liquid ethane. Why we are using liquid ethane, I will come back to this point later, and then it will form this kind of ice. Now, if you just consider, as I mentioned, this is the carbon area. So, I can use, so this is a carbon area. When we are using, if we want to focus our electron beam, so if we just take this particular case, that okay, this is a carbon area, and then there is a ice formation here. The ice is called a amorphous ice. This is a very soft ice. Then if we want to focus for any imaging, there will be two things. One will be exposure. Exposures means when we are giving certain dose and collecting the data, and another one is that you have to do the focus. Now, if this area is an amorphous ice and protein is embedded here, and if I want to focus in this particular area, what is going to happen? Your electron dose has a high energy, it will just started melting this entire ice, and it will give a thermal drift also, and your sample will be exposed before you collect the data during your focus. So, that is a problem. That is the reason we cannot focus within this ice. What we have to do, we have to shift the electron beam in this particular position, where we will focus in this carbon area, where we do not have the too much amorphous ice. We can easily do focus here and then come back here and collect data here. So, whenever we are collecting data in this particular area, this area is not exposed before we collect data. That is the reason whenever we will do the focusing for the cryo-electron microscopy, we are focusing in a different place, and then we come back to this particular area and collect data. So, our before imaging, our sample imaging area should not be exposed by high energy electron beam, and the second one is that it should not damage the amorphous ice and should not create any thermal drip in that particular region. Now coming back to the focus, if you remember last class we are discussing that okay, when there is a electron beams are there, electron beams can converge in one particular area. So just consider this is the actual focus. Okay, now there is a other two possibilities is there. I am changing the magnetic field in such a way there is a crossover point somewhere up. This is called over focused, that we discussed last class. Now if we just do the other one, that okay, we have an electron beam and we are using a magnetic field in such a way that electron beam come here and merge somewhere here, that is called the under focused. So what is the difference between focused, over focused and under focused images? It is a very important part now. So always remember that if images are focused, our eye is not capable to see that focused image clearly. If images are over focused or under focused, it can create a very nice image of that particular sample. How this is happening, I will come back and I will show that thing with a video. So before that, let me explain little bit more with a ray diagram again. So you notice that the focused image, what we are getting, focused image, electron beams are focused here. So when I was talking about that, okay, my eye can see when the sample is over focused. This is the focused image. Okay, my eye can see it very clearly. What does it mean? So if you just consider that, okay, if we have something that, okay, I will just take a very light color here because my background is very light. So now I want to draw a duck here, and here you just remember that I am taking this intentionally very light color here because my background is white and then I am taking a very light color, so you cannot see it very clearly. Now if I make a background outside this duck, then it will be very clear to you because there is the outside background. So when you are just focused your beam, that time you do not have any background like that. It is only that your this yellow color duck, you can see it, that is coming here, it is giving like this one. So right now whenever I am talking about this is a under focused or over focused image, that means when there is a over focused, electron beam is converged somewhere here above the image plane. This is over focused. When this is under focused, electron beam is converged somewhere below the plane. This is under focused. Now most of the time there is a over focused image, there will be a black. So I will just redraw that. Okay, so there, if this is my duck, you consider that, okay, this is the duck I have, and now it has a 3D structure here more and more. Now this particular part will be very clear when I have a black background. So when this is a over focused image, what will happen, you can see outside of your particle or outside of your image there is a black fringe outside the sample, and that will help us to see the sample. And when there is a under focus something, that if I have a dark as a black, so this is my sample. So you consider that this is my duck, it is the tail, and there is a outer line, something white outer line you can find out outside the sample, that will help us to see the sample and separate it from the background, and it will be easy to see when it is separated out the background. That is the reason most of the time we prefer over focused or under focused images, not the focused images. Focused images, it is very difficult to see the sample. Now take a look about the signal. When we are transferring the signal, what kind of changes it is happening of the signal. So when there is a focused image, if you just put the spatial frequency here, and if this is the amplitude, what it will happen, you can see there is a electron beam. When it is under focused, electron beam is going like that. When there is a focus something, either it is over focused or under focused, so it will either go in this direction or this direction, but there we can see the waves is oscillating more. Now if we are using more and more focus, this oscillation will be much higher, so it will go like that. So this ray diagram, we can convert these things to a sine and cosine curve to describe that how the signal is transferred from one form to other form. That we are going to discuss more. At this moment, let us take a look in an actual scenario when we have a sample, what I am talking about, the black fringe and white fringe, or how we can distinguish those samples from the background. So this is very important. One more thing you should remember, that last class I also discussed about the astigmatism. Astigmatism is a very common problem whenever you will go to the microscopy room. If there is a unstable lens system, or if there is a little bit variation in the magnetic field of the lens, what will happen, there will be electron beam will be converged in a two different plane. From one side of the lens is a asymmetric, so one side it will converge in a one plane and other side it will converge in another plane, and then it will give you a elliptical kind of power spectrum. How this power spectrum should look like, that we are going to discuss. So let us take a look how those images are looking actually. Okay, so now I will play video here and I hope you can see it. Let me explain these things little bit. So here what you can see a power spectrum and a very low defocus. That was I was talking about, that when you have a very low defocus, you cannot see the power spectrum as well as the image. So let me first discuss about a different defocus value and the astigmatism. Now if you come to this particular point, you can see here that at a very low defocus the power spectrum is completely invisible, and even if we are going to particular defocus value, you can see it is a elliptical either in x axis or y axis. So definitely there is a astigmatism. If we have to figure out if there is any astigmatism or not, whenever we are collecting data, it is better to correct this one. So in those particular cases, we look at this power spectrum and try to correct it. Most of the time whenever we have to correct the astigmatism, we have to use the multifunction x and y lenses. If you remember, the multifunction x and y lens is made by quadruple lens system, where there is a north south is connected with each other. So that we discussed during the electromagnetic, when we discuss about the electromagnetic lens system. Now you can see there is a elliptical power spectrum, and we are gradually correcting this one and trying to make it circle. So if we just wait little bit, now you can see this is the power spectrum is almost circle now. So that is the an important point. Whenever we will go to microscope room, we will look at the power spectrum at low defocus value, because then low defocus value, it will really help us to see the beam is circular or it is elliptical. I forgot to tell you one thing, for the cryo-electron microscope or TEM study, most of the time we are using under focused images, and that is we are calling defocused image. So, I will use this defocused term very frequently. Whenever I will use this defocused term, you can consider that it is a minus focus or under focused image, not the over focused image, because over focused image will get a black boundary and particles will appear as white, and that will be a problem when you will do the data processing. That is the reason most of the time we will use under focused image that we call the defocus. So, when we will have a very low defocus, you can see the beam is very big, spreaded all over the screen, and it is a very elliptical beam. That time it will be easier to see is this beam is really elliptical or not. If we are using a very high defocus, this power spectrum will be very centered. So, most of the signal will come near the center, and that is the time it will be difficult sometime if your eye is not very trained, it will be difficult to see the astigmatism. That is the reason one of the suggestion is that you go low defocus where you can see this elliptical beam, and then if you can see the elliptical beam, there is a multifunction x and y will be there near the control panel. You have to use very gently the multifunction x and y to correct the astigmatism. Now, I am coming to the next one, that is very important, that we discuss about that what kind of fringe we can see it when we have a different types of defocus value. That is, I told at the 0 defocus value you cannot see your image clearly. 0 defocus value means it is actual focus, but actual focus our eye cannot see it clearly, because there will be no white fringe or black fringe. That is one of the reason we do not collect data at the focus, or even light microscope also whenever we will image, we do not use that focused image, because our eye cannot see it, and then we will say that either it is off focused or contrast is not good. So, that is the reason most of the time it does not matter if we are collecting a data in the optical microscope or not, we are just go below that focus, either below or above, to collect the data. Let us take a look. So, here there is a image. So, in this particular case you pay attention to this one, and this is the power spectrum. This is basically actual sample. Now, I am gradually increasing the focus, and you can see, if you just look at this boundary, you can see a white fringe outside this particle, and that is basically helping us to see this particle clearly. Even those small things, small objects, you can see outside there is a boundary, and most of the cases this boundary is white boundary. That is basically helping us to distinguish the background as well as the sample. Now, this is the power spectrum. You can see we are using a very high defocus, which is helping to see the molecule where the defocus is high and white boundary you can see. Now, we will gradually go in a opposite direction. So, we will reach to the defocus 0, that means actual focus, and then we will use the over focused. And if you pay attention to this particular area, you can see a black boundary outside of this sample, and so now you can see it is almost 0, where the boundaries are not visible. So, sample is not clearly visible. Now, if I go gradually to a different defocus value, you can see a black fringe outside the sample, which is here also black fringe is visible, here also black fringe is visible. That is basically helping me to distinguish this sample. So, either it will be white fringe or it will be a black fringe, and that is basically helping us to distinguish the sample from the background. And for the biological science sample, it is very important, because most of the time biological science sample will stay inside a buffer solution, which is basically either phosphate buffer or tris buffer or Hips buffer. And now, component of those things are very similar to component of the protein, DNA or RNA, and it will be carbon, hydrogen, nitrogen, oxygen, maximum it will be sulfur, phosphate and some metal ion, and you can see it is a not that heavy group, and that is the reason the scattering will be so low. So, then if we really want to see our biological science sample, we have to defocus our images, and here you can see this is not a defocused image, but when you will defocus that image, boundary will be very clear of this big object. even it is not a biological science sample. Also one important part is that whenever we under focused some images, so as I mentioned that if this is the plane, where if you just consider this near my nose is the image plane, and your electron beams are coming, under focused image is converged somewhere here, okay, and this is actually image plane. So and the over focused is also going above the image plane. So in these three cases, as I mentioned that focused, it will be really difficult to see any object or particle. Under focused and over focused images are good, but also I mentioned that if you look at those black spot carefully, I will play this video one more time, then you can notice that you know that over focused image is looking very different than under focused image. That is the reason if we are collecting under focused image, we have to be consistent and we have to collect most of the data in an under focused condition. If we want to go, generally we are not using over focused image, mainly we are using under focused image. I play this video one more time, so you can see what I am talking. You have to pay attention to those small molecules here. So this is almost 0 defocus. Now slowly I am going under focused images. Under focused images you can see white fringe, and this is my particles look like. Now from the power spectrum can either, if you just rotate the focus button in anti-clockwise and clockwise, you will get under focused and over focused image. So looking at the power spectrum, you cannot tell it is an under focused image or over focused image. You have to look at the image. So gradually I am increasing the higher defocus where white fringe is there, that means under focused image, and your particle is looking something like that. Now when you will go to the opposite direction, you can see that overall pattern is looking little bit different. What kind of different we can see? Now we are going from here to 0 defocus, and then we will go to the positive defocus, and where we can see the black fringe. Now this is exactly 0 defocus, all the fringes are gone. Now we are going to the opposite direction where we can see the black fringe. Also look at those particles. So these particles is looking quite different here now. So if you just compare with the previous one and this one, overall shape is looking different, overall appearance it is looking different. This is a material science sample. So even if you pay attention to those parts also, you can feel that differences, that okay, the molecule is looking completely different. So that is the reason whenever we are collecting data in a over focused or under focused, we should be consistent, we collect data in that condition all the time. So now we want to compare these things side by side. One is under focused image and one is over focused image. Same image, same position, how it is look like. So if you come here, this is a cryo image. So cryo image, one more condition you should remember. I discussed little bit about the cryo-electron microscopy. Cryo-electron microscopy, we are not using any staining solution. Your sample will stay in a normal buffer solution, and it is a phosphate buffer or Tris buffer or Hips buffer, and your contrast between the sample and the protein contrast between the sample or protein sample, whatever you consider, with your buffer, it is almost same. So it will be very difficult to see if we image it in focus. So I will show you in the later stage when we will talk about the sample preparation. If something is focused, it is bit difficult, but sometime for 3D reconstruction purposes it is good. So generally high defocus is good for our eye, because there is a very thick white fringe and it will help us to see the molecules properly, but sometime it is also restrict our high frequency signal or high resolution signal. That will come back later stage and discuss. So here you can see those black molecules is my protein sample here. Those are the protein sample here I am pointing. So all over this is full of the protein sample. Why this sample is looking black? As I mentioned, in cryo-electron microscope our sample will stay inside the buffer, and buffer and protein, if you just compare, protein is much denser than the buffer, and it has all this molecule and is completely packed, and when there will be a scattering, electron can go through easily through the ice or buffer, but not through the protein. Scattering will be more there, and that is the reason our protein appears black in a comparatively white background. But this is the under focused condition we are collecting data. Now same data I want to collect in a over focused condition. Whenever I will collect the same image in a over focused condition, you can see this entire image is looking completely different here. There is, I cannot recognize my protein at all. So if you are collecting suddenly that your sample in a over focused condition, protein will appear in a completely different way, and the contrast it will inverted. It is not only that, so these are the individual protein molecules here, but if you look at those are the appearing black, where those proteins are appearing as white, as well as there are some circular things are here, this one that appears like this. So there are lots of protein molecules are there, but their contrast is inverted. So that is the problem is over focused and under focused image, and for the cryo-electron microscope we are interested about the 3D structure determination, right. So that is the one of the major reason we have to be consistent. We cannot mix and match of this over focused and under focused image. So it will be a really problematic when you will do the data processing. Not only the inverted contrast, it is going to affect the signal related part also, that will come back later. So this is all about that whenever we are collecting data at a different defocus value and what kind of focus we are using. So you remember one thing, this is very important, that you should correct the astigmatism or operator whoever is using it, does not matter it is a negative staining microscope or cryo-electron microscope, you have to correct the astigmatism before you collect the data, and this astigmatism may come from the condenser lens as well as the objective lens. If required, both the things you have to correct. Also it will be always good to use some kind of defocus value. So generally for the biological science sample we are using the under focused image, and the focus button or objective lens current we have a focus, that is you can rotate, it is just a circular button which you can rotate in a clockwise or anti-clockwise, which will give you a under focused and over focused image. Based on your requirement you can play around with those focus value. Now next class I am going to talk about more about the pump as well as the detector. Detector will spend more time because that is connected with our actual data. Pump is not that important for the common student, but it is good to know. And thank you all. I will finish it here, and thank you for your attention.